So for those of you who don't know me, I'm Agnieszka Carpenter and I'm the Executive Director of BioME. And I wanted to welcome everybody to our 2021 BioME Student Showcase, the college edition. First, let me thank all of our sponsors. These are our annual sponsors, and without them, we wouldn't be able to do what we're doing right now. And so the way it's going to work today is every contestant will have 3 minutes to give a pitch and talk about their research. And then you have about 5 slides. And then after the 3-minute pitch, we will have some questions from the judges. We ask that everybody stays on mute unless it's your turn to talk. I will be calling you by name. We have 4 judges. And I wanted just to introduce you the judges. Next slide, okay. So we have Dr. Casey Doucette from IDEX, Gary Goodrich, Replanova Farm, also BioME board. All of our judges are on our BioME board. Patrick Breeding, Marin Skincare, and Bethany Fortier from IDEX. And so these judges will be asking you questions. And then here is the order of appearance for today's presentations. I think all of our contestants are aware when your turn is. Just please be ready. When it is your turn, I will call you by name and I will ask that you share your screen. And so then we'll give you the 3 minutes. And then when we are done, when all the questions are asked, we just ask that you stop your share. Yeah, and I think we're going to jump right into it. I know some of you are doing it for the second or third time, so welcome back. And for all of our new contestants, welcome and good luck to all of you. Also, I wanted to just welcome all of our guests. I hope you enjoyed today's presentations. It's really fun, and we're once again wishing good luck to all of our finalists today. And with that, our first contestant today is Zaid Al-Abbasi. I also wanted to say that if I butcher anybody's name, sincere apologies. So maybe let's do this. When it's your turn, I would like for you to introduce yourself, tell us the title of your project, and then the floor is yours. Okay, so Zaid?
Yes.
Hi.
Hi.
Am I saying your name right?
I think so, yeah. Okay.
Yes, so whenever you're ready, you can share your screen.
I think I can. Okay, let's see. Do you see this?
We can see this. Okay, so.
Okay. So I'll start.
Yes,
okay,
whenever
so,
you're ready.
Yeah. So chronic pain is challenging to treat and maintained by neuronal plasticity in peripheral nervous system, PNS, and central nervous system, CNS. My name is Zaid Al-Abbasi, and I am a first-year GSBSc PhD student. Today, I will be guiding you through my project examining the role of CAMP signaling pathway regulators in acute to chronic pain transition. So, my interest in this project comes from my passion to investigate how cell signaling impacts sensory system and pain specifically. And this project employs my passion with purpose of exploring the targets of transition from acute to chronic pain. And this project has a potential to have a major impact as chronic pain is an epidemic across the US and the globe, tens of thousands of deaths due to opioid overdose yearly, and this work will help to identify effective chronic pain therapeutics. Cyclic AMP activation produces hyperalgesia and an increase in sensitivity to noxious stimuli, to noxious thermal or mechanical stimuli, and CAMP inhibition reduces hyperalgesia in inflammatory and other pain models. Hyperalgesia plays a critical role in acute to chronic pain transition, and proteins mediating CAMP signaling are organized into complexes by scaffold proteins called A kinase anchoring proteins, or ACAPs, such as ACAP12. And global phosphoproteomic analysis done in the Molliver lab for dorsal root ganglia collected from mice inflamed using prostaglandin E2 injection showed a significant decrease in phosphorylation of ACAP12. So we run a conventional PCR for multiple tissues from normal mice and rats, and the results show that most of tissues in rat and mouse have a specific dominant isoform of ACAB12, while in mouse DRG, the bands indicate relatively similar abundance of all these three isoforms. This finding suggests that an isoform-specific complex formation and possibly different functions among the isoforms. Consequently, we designed an experiment for more understanding of the response of ACAB12 to inflammation, and the results showed a varied expression pattern of these three isoforms. So in conclusion, based on current results, we are confident to propose that there are three ACAB12 isoforms in mouse, not only two. Based on microdomains concept, that means each one of these isoforms could have specific location inside the cell to function. And ACAB12 is one of the novel targets focused on recently in cancer and cardiac field. And these findings open in a new avenue to understand ACAB12 role, not only in chronic pain model, but in other fields as well. So that's what I have, and thank you very much.
Thank you so much. Great job. And now I'm going to open it up for the judges.
I can ask the first question. This is Casey. I was just wondering how, if you could maybe hypothesize on how you would target ACAB12 and maybe touch upon how the cancer and cardiac fields are looking at that and what they're doing there.
So most of the papers I read about the cardiac and cancer field, they used microRNAs to target these isoforms. Like there is for ACAB12 beta, they use the specific mRNA, and ACAB12 alpha, it's regulated by DNA methylation. And based on these techniques, they are working to identify the rules of each isoform of this protein.
Great, thank you.
Yeah.
Do we have another question?
Yes, I have a question.
Yes.
Based on this work, you say that you're looking for specific locations to function. What are your future plans on determining how to do that?
Actually, we have a caveat because the antibody is still not working well on isoforms specifically, but we're trying to mute one of these isoforms and try to run like immunofluorescence and see the locations of other isoforms. And based on that, we can work more on identifying the micro domain of each isoform.
I see.
But we still have to, maybe we need to do a new antibody because the antibody is only for generic ACAB12.
Right. Okay. So antibody development is on your list.
Yes.
Thank you.
You're welcome.
Okay. Thank you so much, Saeed. If you can stop your share, that would be great.
Okay.
Because thank you so much once again. And now we're going to move on to our next contestant, and that's Khalil Barai. Please correct me.
Okay.
Hi.
Hi, can you hear me?
We can hear you, yes.
Let me share my screen. Okay, can you see my screen right now? Sorry for the interruption.
We can. We are seeing the presenter's view. I think it's fine, but you might want to switch it to.
Okay, can you right now see the whole?
Now
Okay, great. Sorry.
No problem. So whenever you're ready.
Yeah. Hello everyone, I am Kalal. Welcome to my presentation. I am a master's student at University of Maine. Today I'm going to present my research on UV-based remote sensing platform to detect the spatial variation of wild blueberries for precision agriculture. So wild blueberries are one of the major crops in Maine. Two main factors for driving the yield of wild blueberries are irrigation and fertilization. But current conventional management practices such as irrigation and fertilization treat the field uniformly. But soil water retention capability and soil nitrogen availability differ significantly across the field due to the variation in soil physical and chemical properties. This type of uniform management without considering the special variability of soil properties may lead to result in over or under application of resources within a location. So that may overall result in pollution, reduced yield and profitability, and overall affecting the sustainability. So that is why the blueberry farms need a system to quantify those variabilities and treat the field based on these variabilities. So precision agriculture has the potential to do that. It is a farming management concept based on quantifying and responding to inter-field variability of crop cities. The main ideas of precision agriculture are doing the right management practices, that means irrigation, fertilization in the right place at the right time using new technologies and field data. So for the precision agriculture, to precisely monitor the variability of field, we are using remote sensing tools. So remote sensing is a non-destructive and cost-effective method which has the potential to quantify the spatial variability in real time. In our study, we used UV-based multispectral sensor to collect multispectral imagery of the blueberry canopy. So based on the reflection difference of canopy in different wavebands, different indices are developed. So NDVI has been developed to quantify leaf chlorophyll content, which also tell us about the leaf nitrogen availability, which also tell us about the crop health, and crop water stress index, which is based on the canopy temperature, which also tell us about the real-time crop water stress. So if you look at the image, so on the left on this side, the NDVI, higher NDVI means healthy vegetation. On the right side, the lower NDVI means the stress vegetation. So we did ground sampling for leaf water potential for validating crop water stress index and leaf chlorophyll content for validating NDVI as an indicator of leaf nitrogen status. So if you look at the picture, despite having same type of crop inputs, that means irrigation for here, some of the parts of the fields are more stressed compared to other parts of the field. This is also true for the nitrogen availability. So overall, this result suggests that the special variation of crop water stress and leaf chlorophyll content within the field can be successfully mapped using crop water stress and NDVI indices. So this special variability suggests that the need for precision agriculture. With precision agriculture, fields can be divided into multiple management zones for customized management inputs based on the special variability. And adaptation by irrigation precision agriculture can help the wild blueberry farms to reduce nitrogen leaching and pollution, increase overall water use efficiency, increase the yield and profitability, and overall it can help with the sustainability of agriculture production. So these are all the references I used for these slides, and thank you.
Thank you so much. That's fascinating. And now some questions.
I have a question. Good presentation, Kalal.
Thank you.
I was just wondering how you would think about detecting the accuracy of healthy versus stressed, and what type of factors might contribute to background noise that you might want to consider.
So for the current methods of monitoring the water stress, those are proven to be accurate. So to validating the indices, we are comparing with the field-based data, field-based sample. For example, for validating the crop water stress index, we are doing regression models with the leaf water potential. So leaf water potential is a is a already validated method for monitoring the leaf water stress or crop water stress. So by doing the regression models, we are validating how effective crop water stress index or NDVI to remotely monitor the overall health of wild blueberries.
Gotcha. Thank you.
I have a question. Can you talk about sort of the reproducibility of your measurements?
Yeah, so right now, so that project is already ongoing. So we collected in 2020 like 5 times, and we have also planned to collect the data on 2021 this summer. So we are comparing in different development phase of wild blueberries to figure out if the method is suitable for in different development phase of wild blueberries, to see that if it's like reproducible or not.
Thank you.
Okay, thank you so much, Khalil. If you could stop your share, that would be amazing. And then
Thank
we're
you
gonna
all.
move on. Thank you so much. We're gonna move on to our third contestant, and that's Avery Bond. Hi, Avery.
Hi, let me pull up my screen. Can you
Awesome.
hear me okay?
Yes, we can.
Okay.
Okay, and whenever you're ready.
Okay, can you see full screen mode and everything?
Yes.
Okay, awesome. Okay, so hello everyone. My name is Avery Bond and I graduated from the University of New England in 2019. I am currently a second-year PhD student at the University of Maine in the McGinnis Lab, where we study virus-host interactions. So specifically, we are interested in learning how to prevent JC polyomavirus from causing deadly disease in humans. So JC polyomavirus infects 80% of the global population. So about 4 out of 5 of us listening right now are actually infected by JC. In healthy individuals, JC virus infection is an asymptomatic infection of the kidneys. However, in immunocompromised individuals, JC infection spreads from the kidneys to the brain, where it causes the disease progressive multifocal leukoencephalopathy, or PML. PML results in the formation of lesions in the brain caused by JC lysine glial cells. Unfortunately, deterioration is very rapid and death often occurs within 1 year of symptom onset. So although JC virus was discovered about 50 years ago, there are still no effective treatments. So in an attempt to identify treatments for JC polyomavirus infection and PML, our lab conducted a wide-scale drug screen of over 700 drugs from the NIH clinical collection. Since these drugs are already in development for other uses, this method of repurposing drugs is an efficient means for antiviral drug discovery. And this has actually been done recently to identify remdesivir as an effective antiviral treatment for COVID-19. So we tested the drugs in the NIH clinical collection to determine whether they had inhibitory effects on JC virus infection. So we identified 43 hits and further characterized them into drug classes shown in the pie chart. So drugs that impacted calcium signaling were an attractive target due to preliminary evidence gathered in the McGinnis Lab. And so this graph here is a quantitative representation of all drugs included in the screen, with yellow circles representing calcium-related drugs. And this table here shows all of those calcium-related drug hits, and then the drugs in red text at the bottom were further explored. So we are currently working to further validate and characterize some of the calcium-related drug hits to identify how these drugs might be reducing JC infection. And so utilizing an in-cell Western approach, we found that nifedipine, an L-type calcium channel inhibitor, significantly reduced JC infection in human embryonic kidney cells. Additionally, TFP, an inhibitor of calmodulin, also significantly reduced JC infection in HEK cells or kidney cells. These results suggest that calcium signaling pathways might be important for JC infection in kidney cells, and also that to establish drugs reduce JC infection in vitro. So in conclusion, 2 drugs previously approved for other uses, nifedipine and TFP, reduced JC infection in kidney cells. Our next steps include investigating the mechanism of JC inhibition during the viral infectious cycle and determining if there are cell type dependent differences in pathways utilized by JC. Thank you to our funding sources, and thank you all for listening.
Thank you so much, Avery. I've never heard of JC infection. Now it's another virus I'll be scared of. It's up for the judges now to ask questions.
I have a question. Just a quick one about the frequency of the lytic brain activity. I mean, you said it 80%, but what's the frequency of the more serious lytic effects?
Right. So somewhere between, about 40 to 90% of the global population is infected by JC polyomavirus. And like I said, it's usually an asymptomatic infection, and it kind of just hangs out in your kidneys. And as far as we know, doesn't harm most of the population. But in severely immunocompromised patients, such as those with HIV or someone with multiple sclerosis that is currently receiving immunomodulatory therapies, that's when we really see PML occur. And that's where that is the lytic infection in the brain. So it's pretty rare, but when it occurs, we don't have any treatments for it.
Okay, thank you. And just one other quick question. I must have missed the model that you did the screening in. What was the model?
We did it in cell culture. So we tested on, yeah.
Okay, thank you.
I have a question. You mentioned that some next steps are to determine cell type dependent differences in the pathways utilized by JC. Can you touch on some of your research that will lead to that?
Yes. So these talks are 3 minutes, so it's kind of hard to fit in all of our results, of course. So we test infection in kidney cells and brain cells. And so what our findings in kidney cells may not necessarily translate to what we find in brain cells. And so our future directions are to really flesh out and characterize what's going on in kidney and brain cells. And I currently work in immortalized cell lines, but moving forward, we'd like to also test in primary cell lines.
Great, thank you.
And along those same lines, do you have information from the drugs that were in the screen, or do you intend to collect this data on whether or not there are effects on bone of these drugs?
Effects on bone?
On bone, yeah.
I guess, is there a way you can rephrase your question?
Yeah, I guess I'm just wondering if there's any information on the drugs that you used and whether they have like inhibitory or sort of the opposite effects on bone growth. Or maybe that would be, I think, because if you're sort of targeting the calcium channels, I'm just wondering if that would be something that you might look at as well.
I see what you're saying now. That is something I haven't investigated. So the 2 drugs that I included here that have reduced JC infection, one is nifedipine, and that is currently, interestingly, used to treat angina, so chest pain, and then also it's used to treat hypertension, high blood pressure, and then TFP, the other drug, is currently used to treat schizophrenia and then also sometimes for anxiety, but yep.
Thank you.
Mm-hmm.
Okay, thank you so much, Avery. Thank you, judges. Avery, if you can stop your share, thank you so much. And now we're moving to our 4th contestant today. We are going through all of our contestants. Our next one is Ketty Cormier.
Yep, hello. All right, I'm gonna check my screen. Okay, can everyone see that okay?
Yes.
Great. All right. Hello, my name is Kitty Rose Cormier. I'm currently a junior studying civil and environmental engineering at the University of Maine. I'd like to thank the Bioscience Association Maine Undergraduate Showcase for inviting me to talk today. And today I'll be talking to you about SARS-CoV-2 testing in wastewater. So what is the problem? Unfortunately, it's one we know all too well. So currently in the United States, over 32.3 million people have been infected by SARS-CoV-2, and over 500,000 people have lost their lives. The SARS-CoV-2 pandemic has also had a disastrous impact socially and economically for people all around the world. There's a large need for small rural communities, colleges, businesses here in Maine to open up to allow for economic relief without the implications of invasive and expensive sampling methods. So I'll talk to you about my areas of research. So since humans shed SARS-CoV-2 in their feces, wastewater-based epidemiology, also known as WBE, is a vital tool that can be used for health monitoring. This form of testing has the advantage to integrate the results of an entire community so it's anonymous and less invasive than testing all individuals. Routine wastewater monitoring would enable small towns and businesses to increase restrictions when the virus is circulating in the community. Current commercialized testing kits are priced high and require expensive equipment that most wastewater treatment facilities cannot afford nor have access to. The goal of the research is develop cheaper, effective alternatives to create a complete wastewater testing kit that can be used at wastewater treatment plants, colleges, and more. So this is actually an example of some containers that we've used. So we've done testing at UMO, UNE, and USM. So now I'll talk to you briefly about the methods. So wastewater that is collected at a wastewater treatment plant or at collection sites outside of residential buildings is the combination of the entire population's water. So concentration and extraction methods are necessary in order to capture the accurate amount of virus shed by people infected with SARS-CoV-2 within the community's population. So detection methods are used to identify the quantitative or qualitative presence of SARS-CoV-2. Now I'll show you some pictures. So for collection, I researched the automatic sampler collection method as well as cotton swab sampling. We were donated tampons by P&G, and we made a simple little mechanism using a carabiner and a nylon string and hung that in a manhole. The next step is concentration. So we actually used an IDEX kit, hello judges, the PEG 8000 kit, and then we also used the NanoTrap magnetic bead kit. For extraction, we also used another IDEX kit for magnetic beads, and we also compared it to a simpler treatment of heat treatment and centrifugation to extract nucleic acid from the virus. And lastly, we used detection. So we researched RT-LAMP and qPCR testing. So for the sake of time, I'm not going to talk too in depth of all of the different processes, but I'll focus mainly on the collection and concentration steps. So here are some nice graphs. Now these are comparing the cotton swab collection method to the automatic sampler method from Somerset Hall and the UMaine North Line. And as you can see, the cost of equipment and approximately time required for the cotton swab sampling method versus the automatic sampler is large. So for cotton swab sampling, it only costs around $20 to $40 to sample at all 6 sites on campus. With the automatic sampler, we bought them used, so we were actually saving money, but that still cost around $3,000 a sampler. So quite a bit of money. Now for the time, it takes approximately 2 minutes to set up a cotton swab sample, and it can take up to an hour for setup and collection for the automatic sampler, and can take another hour just for cleaning and maintenance. So for the concentration method, we compared the different concentrations for PEG 8000 and the NanoTrap. And the cost of equipment used for these samples were also very large. So using a temperature controlled centrifuge varies from $5,900 to over $6,000. And the approximate time to run a PEG 8000 concentration method on one wastewater sample can take up to 6 hours versus the series NanoTrap, which only uses a magnetic frac, which is around $60 to $90 and only takes 2 hours. So in conclusion, obviously I didn't have time to talk about the differences in extraction as well as detection, but some conclusions that we can draw from collection and concentration is that the most promising concentration method to use directly at a wastewater treatment facility is the series NanoTrap concentration method in comparison of equipment needed, time, and feasibility of testing multiple samples. The PEG 8000 concentration method does use more expensive equipment and takes approximately 3 times longer to complete. And the cotton swab sampling method shows promise for simpler collection in manholes and outside of dorm buildings and at wastewater facilities. So this form of method is cheaper, simpler, and takes less maintenance time than the automatic sampling method. Now, these results show the possibility for commercial development of a cost-effective testing kit that wastewater treatment plants can use without having to pay for expensive lab fees or equipment. And these are my sources, and I'd like to thank you for letting me talk today. I'll open the door for questions.
Perfect. Thank you, Katie. Yeah, so whoever's ready for a question.
That's a great presentation, Katie. I noticed at the end you were talking a little about, or at least alluding to, detection sensitivity. Mm-hmm. What are your thoughts on that and the changes in sensitivity between the 2 methods and then compared to existing methods of testing?
Right. So the goal of my testing is more to develop something that's cost effective. So that way you can actually do testing at wastewater treatment facilities. Now, the problem is that people are paying lots of money to send samples to labs. So with RT-LAMP, what's really interesting about it is it's simply a colorimetric method. So it's a simple color change to determine if COVID is present. And that's what's really interesting. And we want to bring that to a wastewater treatment facility because it's less important about high amounts and whether or not it's there at all. So with qPCR, it's more of a quantitative, so we actually get a numerical value. It is like more exact, so that is good for this type of research where we're trying to compare the methods. But it's really interesting. We have to do further research on RT-LAMP and how effective it is in comparison, but it's really cool stuff.
Yeah, that makes sense. And did you do any research into the virus survival in wastewater when thinking about developing a cost-effective method to measure this? Is there a need for it?
Right. So depending on the method, actually, so like with PEG 8000 in concentration, it actually captures dead and alive virus simply because it's just uses coagulation to precipitate it onto a conical, whereas the nanotrap only captures live virus. So it does have some sort of dependency on which one you're going for. But like on campus where we're collecting, it's over 24 hours. So we're more likely to capture live virus as well there.
Gotcha. Thank you.
Mm-hmm.
Any other questions? No? Okay, thank you so much, Ketty. Now we're moving to contestant number 5, a familiar face, Sarah Holbrook. Hi, Sarah.
Hello. All right, let's see. Let's share the screen. And it's working for you all?
Yeah, and whenever you're ready.
Perfect. All right. Hi all, I'm Sarah Holbrook. I'm a BioME graduate student completing my degree at the Jackson Laboratory. I work in the Cox lab where we characterize a whole slew of mouse models, representing different motor neuron diseases, or MNDs, with varying severities and genetic origins. My work focuses on spinal muscular atrophy of respiratory distress, or SMARD, in one of our most severe mouse models, M3. SMARD is a rare, devastating disease that results in early distal neuromuscular atrophy in the limbs and also diaphragmatic paralysis, typically within the first year of life. It is caused by recessive mutations in the helicase gene IGH-MBB2. This causes degeneration of the alpha motor neurons in the spinal cord. Although we have seen significant improvement in muscle strength with some of our gene therapy vectors, as you can see in these cross sections of the leg muscles, it hasn't resulted in 100% recovery in our most severe models, even with the earliest intervention possible. Well, without going in utero. This is important because most of our human patients don't know they have SMARD until they're already presenting symptoms. So how can we make it better? So let me go back to the beginning. Back in 2017, the lab collected the spinal cords of several of our SMARD mice. When we got the results back, we found that several genes in the RIG-I-like receptor pathway, or RLR pathway, were highly upregulated. This at first confused us because the RLR pathway is an innate immune system pathway. But as we looked further, we saw what the RLR pathway detects, double-stranded RNA. Now, not a lot is known about IGH-MBB2, but we do know that it's a helicase. Perhaps the IGH-MBB2 substrate is not being unwound and thus causes an immune response. But wait. The plot thickens. More RNA-seq information from another of our recessive MND models associated with the ribosome quality control gene, NEMF, came in and changed everything. It had the same immune system signatures as our IGH-MBB2 models. And when we bred mice to have one bad copy of NEMF and one bad copy of IGH-MBB2, both which separately have the respective heterozygous phenotype, we saw an extreme MND phenotype with decreased body weight on the left and decreased lifespan on the right, suggesting that these genes are in the same pathway and therefore may have the same disease mechanism. What's the big point? IGH-MBB2 gene therapy works for IGH-MBB2 mutations, but what about the NEMF-associated diseases and other genes that might be associated with those? The RNA-seq data suggests there is a whole new category of MNDs. So the next step is to figure out what role the RLR pathway has in IGH-MBB2 and NEMF by breeding knockouts of several key RLR players.
This is essential for developing therapeutics that would not only target IgH MBP2 MNDs, but a whole new unexplored category of RIG-I-like receptor-associated motor neuron diseases. Thanks, and for more information about the concepts, you can follow these links.
Thank you, Sarah. And now I'm going to open it up to the judges.
I have a question for you, Sarah. You mentioned creating knockouts. That's a lot of knockouts that you're looking at there. Do you have another method that you want to follow up with to sort of maybe go about a knockout in another way?
Yeah, there's so the main point of the knockouts is just to see if the RLL pathway is beneficial, if it's detrimental, or if it's just reacting to all this double-stranded RNA. These knockouts have already been created, so we just have to breed them onto our mice. So that's good, but there's other ways where we also want to test out some pharmaceuticals and inhibitors. For example, RIP kinase 1 on the left has some pharmaceutical applications that you can target. And then I know there's some RNase L inhibitors, which I predict is probably the pathway. So I predict that the RLR pathway has a detrimental effect, and it's the reason why the motor neurons are dying, because the ultimate cause of the RLR pathway is to destroy infected cells, or what it thinks are infected cells. And it does that mostly through the RNase L pathway, where it causes RNA cleavage and apoptosis. So,
No,
yeah,
thanks, that's great. That answered my question.
I can ask the next question. Have you ever tried, or do you know if others have tried, sort of rescue experiments where you would sort of give IgH MBP2 to those mice and sort of what happens? I don't even know if that exists and could be a treatment, but could you speak to that at all?
No, yeah, I was definitely thinking of doing that for one of our experiments. Like I said, we've been doing gene therapy for IgH MBP2 mice. So we have the virus. We would just have to get permission from our collaborators. But what I'm really interested in is, so I've been reading some papers, and there's one that came out 3 weeks ago, where it associated the RIG-I pathway with ALS and other genes associated with that. And they saw that when, what's the gene called? It's TARDBP, it's TAR binding protein 43. When you deplete that in SOD1 mice, so SOD is the most common form of ALS mutation, they actually saw a betterment in those mice, less of a atrophy. So I kind of answered your question there, but like I said, I feel like there's a whole bigger picture too, and if we can target this pathway, we can connect so many other motor neuron diseases and help out multiple, because the gene therapy, it's working great. Let me see. Yeah, it works fantastic, more than we could have dreamed. But if we can do something that can target some forms of ALS and other rare diseases, then yeah, that's the big goal for me.
Great, thank you.
Thank you, Sarah. That's very promising. If you could stop your share, that'd be awesome. Okay, and so we are halfway through. Maybe because we are halfway through, I just want to mention a couple things. So this session is being recorded. If you want to share it with anybody afterwards, it will be on our YouTube channel. And also, I just wanted to mention the prizes for today's presentations, not to put any pressure on you, right? But the prizes are $2,000 for first place, $1,000 for second, and $500 for third. So it's pretty competitive, and I wish you all good luck once again. And our next contestant, number 6 today, is Bright Obeng.
Right. Can you hear me?
Yes, we can.
Right. Thank you for the opportunity. My name is Bright, a graduate student in the Gauss Lab, Maine, and I present on how the antimicrobial cetylpyridinium chloride, CPC, inhibits immune mast cell function. Mast cells are ubiquitous through the human body, important in immune and nervous systems. They share many signaling elements with numerous other cell types. Mast cells degranulate to release bioactive substances like serotonin and histamine. Over the past decade, our work, together with other scientists, led to the ban of the antimicrobial triclosan from virtually all products. Now we are focused on studying CPC. CPC is used in numerous personal care products, cleaning products, and agricultural processes. Concentrations found in consumer products like mouthwash are above the critical micelle concentration, where they act as a detergent to solubilize microbial cells. There is lack of data on the toxicity of CPC towards eukaryotes, and the goal of my project is to investigate the effect of CPC on immune cell signaling. There is the need to provide data so that safe exposure levels can be set for humans and also for wildlife. We hypothesize that the positively charged CPC electrostatically targets the negatively charged PIP2, leading to the suppression of antigen-stimulated calcium influx, leading subsequently to the inhibition of degranulation. There is evidence that PIP2 is electrostatically shielded by cations and the antibiotic neomycin. Using a fluorescence-based assay, we discovered that CPC inhibits degranulation at a concentration as low as 1 micromolar, which is 1,000-fold lower than concentrations found in consumer products. CPC also inhibits the influx of calcium by 40%. Using confocal microscopy, we discovered that CPC traps PIP2 within the plasma membrane, making less PIP2 available for signaling. CPC also suppresses the polymerization of microtubules, which are the railroad tracks upon which the granule cargoes are moved to the plasma membrane for exocytosis. In conclusion, our results point to immune cell dysfunction caused by CPC at a concentration which is 1,000-fold lower than concentrations found in consumer products. CPC suppresses the influx of calcium, which is crucial to the degranulation pathway. And it does this by interfering with the availability of PIP2. CPC also suppresses the polymerization of microtubules. This research provides biochemical mechanisms by which CPC, ironically, may undermine the human immune system, and also provides data to aid regulators determine safe levels for the usage of CPC. PIP2 plays a key role in viral infection, so CPC's mechanism of action may suggest helpful antiviral properties of CPC as the state of Maine and the world at large fight the COVID-19 pandemic. Thank you.
Thank you so much, Bright. I definitely learned some new things about mouthwash here. Okay, so thank you for that. And now time for the questions.
I have a question.
Oh, go ahead, Patrick.
Okay, I'll go, sorry. I was just gonna ask, what would you do next to investigate the extent of the impact in vivo that such low concentrations have? You know, in vitro is one thing, but how would you investigate the extent to which it affects, you know, actually in more realistic models to actually demonstrate this effect further?
Yes, so that would be to probably work with a mouse model, but we don't use that. We don't use a mouse model in our lab. However, we have other labs which do that. So every now and then we check up on articles from that lab. For example, when I spoke about the work that we did over the past decade on triclosan, there is a graduate student from the GOS lab here who is now in a government institution, and they work with my mouse or mice model. So when she left here, they are now trying to see the effect of the triclosan within the mouse model. Yes. So what I would do from here is to replicate these experiments in other cells, like the T cells and other cell types. That is what we do. Yes, to see that whatever we are seeing here can actually be replicated in other cell types.
So you're going to check out other immune pathways in vitro.
Exactly.
Gotcha. Thanks.
If I could just follow up on that, you mentioned other cells. Has your lab thought about looking at basophils given their similarity to the mast cells at all?
Come again, I did not hear that.
Sorry, I said following up on your statement about investigating this in other immune cells, has your lab thought about investigating basophils given their similarity to mast cells and their progenitor similarity as well?
Yes, we have actually thought about, yes, using basophils. We consider that T cells as well. We also consider the use of what they call T cells and also primary mast cells from humans as well. This particular model we use is the rat basophilic leukemia cells, RBL, which behave functionally just like the mature human mast cells. So we start from here and we progress to the use of the primary human mast cells and also T cells and the basophils, as you are saying.
And I'm guessing that you're interested in the T cells because of their interaction with the mast cells more as your next step? Or is there another reason?
No, simply because what they call the signaling, the calcium signaling of the mast cell is similar to the T cells. And the degranulation pathway actually revolves around the calcium signaling. It's central to that. So then because there is similar signaling in terms of the calcium, we'd want to just replicate that in the T cells. But then we are not going to limit it only to the T cells. We'll look at other cells.
Perfect, thank you.
Okay.
Thank
I was just gonna, Agnieszka, should we close it out?
Patrick, if you have any comments or questions.
I was just gonna ask, me personally, I find personal care product and cosmetic regulatory testing a very interesting wild west of a regulatory environment. Have you done any research into what companies using CPC have done for safety testing? Have they been very analytical about it, or do you think they should be diving a little bit deeper?
All right, so when you search for data on the toxicity, either from even companies or articles, there is virtually none actually available. In fact, even the mechanism, the metabolism of CPC, its absorption, there is absolutely there's little information on that. So it's just like a new field we are trying to explore, simply because a lot of people are exposed to these things in consumer product. I think everyone here, in one way or the other, might be using a product which has this CPC as an active ingredient. Yeah.
Yeah, that's a good answer. Yeah, thank you.
Thank you. Welcome.
Thank you, Bright. You're scaring me more and more. From now on, I'm only using one.
Don't be scared. We'll crack this code. Don't be scared.
No, I'm aware of it. It's just we don't think about it every day. Okay, so we're gonna now move to our next contestant, and Bright, you can stop your share. So our contestant number 7 today is Prakash Rao.
Yeah, I am just sharing my screen. Can you hear me okay and see my screen?
Yes and yes.
All righty. So yeah, hello everyone. Good afternoon. My name is Prakash Rao. I'm a PhD candidate in the Department of Physics and Astronomy under the supervision of Dr. Sam Hess. And my presentation title is Antimicrobial Agent Cetylpredone chloride interferes with phosphatidyl inositol 4,5 biphosphate protein interactions in influenza infection fibroblast model and in muscle. So Bright just made some background for this presentation as well, I guess. So yeah, let's start. So starting with the motivation, influenza infection and its complication result in the deaths of 250,000 to 500,000 people worldwide. This virus is mutate rapidly and can develop resistance to the drug. So the vaccine needs to be updated continuously. To develop antivirals, we need to understand how the virus interact with the human host cells. And here you can see with the virus structure and with the viral proteins, hemagglutinin HA, matrix protein M2, nuclear export protein NEP, neuraminidase NA, matrix protein M1. So our lab is mostly focused on HA because HA is responsible for attaching the virus to human respiratory cell and infection as well. HA clusters at the plasma membrane, and density of the clusters directly correlate with the infectivity. We also previously showed that the HA clusters are interdependent with the PIP2 clusters. And moving on to the CPC, which is readily available and commonly found in mouthwashes and consumer product. It is positively charged and has been previously shown to have antiviral property in the range of millimolar, which is toxic. So we hypothesize CPC, which is positively charged at lower concentrations, bind to PIP2, which is negatively charged, disrupts HA clustering, and reduces the infectivity of the virus. So the results here are my first results done in collaboration with the Gauss lab. So you can see with the control 5 micromolar and 10 micromolar, CPC disrupts the PIP2 binding protein MUX and releases it from the site from the plasma membrane to the cytoplasm. This is the first instance where we saw CPC disrupts the plasma membrane binding protein, PIP2 binding protein, and we have quantified here. Here the second result over here. Here we use super resolution microscopy technique. It is a Nobel Prize award-winning technique. Here we saw again CPC disrupts the pH domain clustering, which is again a PIP2 binding protein. So we have two instances where we saw CPC disrupts the PIP2 binding protein, and here we saw CPC significantly reduces the HA cluster density. It is important because HA cluster density directly correlates with the infectivity of the virus. Moving on to the next result, we saw CPC disrupts the pH domain and HA co-clustering, which we have quantified here. And here we saw we upgrade our cell model to animal model, and we saw CPC rescue zebrafish with infection. 6 hours post-infection of the post-administration of the infection, 24-hour post administration of the infection, and 48 hours the post administration of the infection. And my conclusions are CPC, which is readily available drug or chemical, is non-cytotoxic at the concentration in the range of micromolar. CPC decreases HA density. This is important because density of HA clusters are more infectious, and also we explain a mechanism for decreasing HA density as well. We also extended our study in the zebrafish whole animal model. And so CPC significantly reduces zebrafish mortality from influenza virus infection, confirming antiviral properties of CPC at lower concentrations. And here are my acknowledgement to all the lab members of ICE Lab, Gauss Lab, King Lab, Magnus Lab, Henry Lab, and Dr. Zimmerberg Lab, and my references. Thank you.
Thank you so much, Prakash. And now I'm going to open it up for the judges.
Prakash, I have a quick question for you. Are you, based on your quick data here, you're suggesting that this antimicrobial agent kills the influenza virus, or I mean, what's the implication here?
Right, CPC at the concentration of millimolar, which is toxic to human cells, kills the influenza virus. Here, we are using concentration at the range of micromolar, 1,000 times less. So we saw, we have a data that is not shown here. It is non-cytotoxic, and it. I'm not saying kill, but we can reduce the infectivity of the virus, and also like we extended that study to our zebrafish model as well.
Okay. Thank you.
Yeah.
Okay, any other questions?
I just had one, Prakash. Can you expand a little bit on the FPOM method that you're using for imaging? Right. It's my understanding this is somewhat time consuming.
Yeah, absolutely. So it's kind of time consuming to put me in the graph. I was limited by 3 minutes. So let me break that up. So every fluorophore or every point-like object makes a point spread functions in the camera, which is of about 300 nanometer, greater than the diffraction limit. Every object we are bound by the diffraction limit. But what FPOM does, what super resolution microscopy does, is that it gets down, it resolves the objects down to 100 nanometer, less than 100 nanometer, and we can see a super resolved image computationally. We do that computationally.
And so how long does it take you to get one complete image?
one complete image with the computational, I'll say for the 10,000 frames, we normally acquire 10,000 frames here in our lab, and I would say 2 hours for that.
Great, thank you.
Yep.
Thank you so much, Prakash. You can stop your share whenever you're ready. Thank you. Okay, so we are moving to our next contestant, and it's Maria Vigna-Lopez. Welcome back, Maria.
Thank you. Can you see my screen?
Yes, we can.
Okay, so hi everyone. Thank you for the opportunity. I'm Maria Vigna-Lopez, and I do undergraduate research at the Wheeler Lab at the University of Maine. For my project, I studied the fungicidal synergistic activity of fluconazole and cyclosporin A on Candida albicans. So first, I want to introduce the fungi Candida albicans and two existing drugs. So Candida albicans is an opportunistic pathogen that can cause a mild yeast infection, but is also capable of getting into the bloodstream and result in severe illnesses, as well as it can be life-threatening in uncompromised individuals. And the current treatment that is being used is the drug fluconazole. So fluconazole is an acyl drug that targets an enzyme in the biosynthesis of ergosterol and leads to membrane stress. And there are two issues that come with the use of this drug. First, it's fungistatic, which means that it's going to stop the growth of Candida, but it's not going to kill it. And the other issue is that it is developing drug resistance in Candida albicans. And then I want to introduce cyclosporin A, which is an immunosuppressive drug that exhibits antifungal activity by inhibiting calcineurin, which is a signaling phosphatase that is part of the fungus stress response. And so the two goals we have for this project is first to understand how the drugs fluconazole and cyclosporin A synergize to be able to treat better the Candida albicans infections. And our other goal is to understand if cyclosporin A works in a similar way to co-culture with Pseudomonas aeruginosa. So our approach is to take different time points and understand when exactly the drugs are synergizing, and also to find the lowest effective concentration of cyclosporin A with fluconazole. In the future, we will be looking at the effect of the interaction of Pseudomonas aeruginosa with fluconazole on the calcineurin pathway, and we also want to study the interaction in vivo using the zebrafish infection model, and we want to be looking at the synergy, the effectiveness of the drug cyclosporin A on the fish, and also the host response to infection. So the project has the potential to help with efficient use of these antifungal drugs to treat the infection. And now I want to go over our results. So here I present on the left graph, I have Candida albicans growth in the presence of cyclosporin A. So this is only the fungi and cyclosporin A. And as you see, independently of the concentration we're adding on cyclosporin A, we see exponential growth of the fungi from 0 to 48 hours, up to a point where there's too many colonies that we can count. However, on the right, the graph represents Candida albicans growth in the presence of both drugs. So we have the fungi, we have cyclosporin A, and we have fluconazole. And as you can see with the different concentrations, from 0 to 24 hours, we're observing death of Candida. And then at 24-hour time points, all Candida was killed. However, we're observing some rescue after those 24 hours. And when looking at the different concentrations, we say that when the concentration is bigger than 3.125 micrograms per milliliter, cyclosporin A alone has no effect on growth. Fluconazole alone only slows the growth. And then almost all the Candida was killed when both drugs are present in 24 hours, and there's rescue of Candida between 24 and 48 hours. And if we're looking at the concentration of cyclosporin, 3.125, we see that it's almost the same effectiveness as the highest concentrations. So that's why in our conclusions, we say that in our in vitro experiments, the presence of cyclosporin A converts fluconazole from fungistatic to fungicidal, and our lowest effective concentration of cyclosporin A is 3.125 micrograms per milliliter in the presence of fluconazole, 12.5 micrograms per milliliter. And then the rescue of Candida after 24 hours shows timeline of synergistic effectiveness. So thank you everyone for listening to my presentation. And I want to acknowledge the Wheeler Lab, Dr. Wheeler, and Dr. Hattab, and of course the University of Maine. And I'll be happy to answer any of your questions.
Thank you, Maria, and whoever is ready for a question. Gary, you're on mute.
Thank you. I have a question. Maria, how do you propose to study the mechanism of synergy in the future and your future studies?
So I'll go back to this slide. So what one of the goals is to see if why is this synergy happening? So how are the drugs coming together to make it happen? So we want to compare, because when we have the fungi with the bacteria Pseudomonas and fluconazole, we observe the same result. So we want to study the two synergies at the same time to see if maybe they have the same mechanism. So we'll start by, in the future, my future step will be looking at the calcineurin pathway in Pseudomonas aeruginosa with fluconazole and comparing the results to what I'm observing right now.
Okay, so that's to see if they're the same, if they work
Yeah.
with the same mechanism.
Yeah.
Okay, thank you.
Thank you.
Do you have any thoughts as to why you were seeing some growth in some of those cultures after 24 hours?
So we aren't so sure yet, and we don't we want to do more concentrations to see if this happens on every concentration, but we are not really sure yet. I think it's also going to be very interesting when we use the in vivo model, because that's with the zebrafish infection model, we can see the live infection. So maybe then we would understand this pattern of growth better.
Follow up on Patrick's question, have you done any observations past that 48 hours and observed what happens then?
No, we haven't looked at after 48 hours yet.
Okay, thank you so much, Maria. Thank you, judges,
Thank you.
for the questions. And thanks for stopping your share. So we have 2 contestants left. Our number 9 contestant is Claire Volk. Hi, Claire.
Hi.
Whenever you're ready.
Okay. Hi, my name is Claire Volk. I am an undergraduate at USM, and I thank you for the opportunity to talk about my ideas about how stable nitrogen isotopes can be used to look at nitrogen cycling in stands of American beech trees, Fagus grandifolia, that are differently affected by beech bark disease. So this idea stems from the acknowledgement that beech bark disease is causing drastic community level alterations to plant communities by decimating between 80 and 90% of Fagus grandifolia trees. And as much as we know that this is causing these big changes to plant communities, we don't know how it's affecting other organisms, in particular soil organisms, and how that might be affecting nitrogen cycling. So in particular, we know that plants expend a huge part of their carbon budgets recruiting mycorrhizal partnerships in order to transfer their carbons for nitrogen compounds. And we know that the abiotic and biotic stressors to plants can also secondarily affect mycorrhizae. So I'm basically developing a project to look at how that relationship might be impacted. This is in the understanding that mycorrhizae are conductors of inter and intraspecific transfers of compounds. So the implications for the specific tree might also impact things at a stand-wide level. So this is based on the ECM model that was developed by Hobie and Colpart. And so as nitrogen moves through the different steps in the process, so certain parts of the process are selective for different isotopes. And in particular, there's fractionation that occurs because of those different selective factors. And Hobie and Colpart and then Hobie and Holberg later on were showing how that fractionation might be related to mycorrhizae, in particular from within the plant tissues itself, from the roots to the stem to the leaves. There are specific ways that's impacted. There's depletion that's going from the roots to the stem to the leaves. And so I'm proposing a project to look at those different signatures and also in the soil. And then in order to see whether those patterns are actually related to mycorrhizae, I would like to quantify the mycorrhizal presence visually, and then look for PLFAs in the soil, which will help give an idea of the biomass of mycorrhizae and fungi in general. And then I'd like to use DNA sequencing with biomarkers like ITS to identify mycorrhizal species that are on the roots to see if there are changes in what those communities look like. And then finally, looking at how the soil nitrogen content is changing. So the different forms of nitrogen would be also analyzed in that, the ammonium nitrate and nitrate. And so I did a very preliminary study for one of my classes where I was looking at the delta N15 values in 2 different stands of trees, one that was showing pretty early signs of beech bark disease and one that was showing later signs of beech bark disease. And then within those stands, I picked a healthy tree and a diseased tree. So this was a very simple factorial model. And what matters is not necessarily the absolute values, but the differences between those values. So you can see that the changes that are occurring on a stand level are much more similar than the changes that are happening between, for instance, the diseased trees in either stand. And then also those patterns of fractionation between, I only looked at roots and leaves, but the fractionation that's occurring in these stands indicates that there could be different patterns of mycorrhizal involvement. So in order to substantiate that, I want to definitely conduct some more replicates and then also try to substantiate the relationship with mycorrhizae. And so this is exciting work because we are still trying to understand the relationship between mycorrhizae and plants and how at a bigger ecosystem level, these relationships could be impacting big nutrient cycling. And stable isotopes might be a way of looking at that. So I'm trying to see if that is an option. And these are my sources. Does anyone have any questions for me?
Any of the judges, just go ahead.
Sure, I have a question for you. So you're proposing to look at nitrate, nitrite, ammonia, quite a bit of things that exist at variability, I would say, depending on many factors. How do you propose you might design your experiment to account for that?
So those are I'm mostly hoping to use those as like covariates in an analysis because the actual signatures that are the differences in signatures that we're seeing like within the vegetation fractionation from like roots to stem to leaves, that's a pretty distinctive signal of mycorrhizal association. So really I'm also trying to quantify to make sure that there's not covariates that are really impacting the values that I'm seeing. And then also it's interesting because as these stands are shifting, there's been studies about how so American beech trees are often replaced by sugar maples in these stands, and that impacts the nitrogen cycling long-term because of the recalcitrance of the leaves, the different recalcitrants, and it changes the nitrogen forms there. So I want to make sure that what I'm seeing in terms of nitrogen fractionation within the plant, it's not due to these long-term changes that are happening. So yeah, I'd analyze it as a covariate.
Great, thank you.
Do you think you might also be able to use this data to predict ahead of time when there might be disruption in nutrient cycling? And if so, is there a different type of data you would collect or different type of analysis you do to try to correlate and project out what might happen?
Yeah, I really think that the model needs to be substantiated before we try to create predictions out of it. I think that there are huge implications for if this could be used as like a diagnostic tool, but I don't really know if it's like reactive or causative. So if for example mycorrhizas are not associating as much with trees that are sick, and that causes increased decline, I think that's a possible thing that we would see, but that first really needs to be substantiated, that relationship between the isotopes can be used as a marker before we try to draw any conclusions from it.
That makes sense. Do you have any thoughts on how you would tease out cause versus correlation? I bet that would be tricky.
I think most experiments, so the experiments that have been done so far have been in a lab setting. They haven't been done in an environmental context. So I think that you could probably pair the work that I'm doing just in terms of creating a correlation with lab studies. And that would probably be a separate experiment.
Got it. Thank you.
Okay, thank you so much, Claire. Thank you, judges. Claire, you can stop your share. And last but not least, we have our 10th contestant today, Michael Wilczyk.
Thank you.
Hi, Michael.
Thank you, everyone. So let me share my screen. Okay. Can everyone see that okay?
Yes.
Okay. And let me revise that. So, hi, everyone. My name is Michael Wilczyk. I am a graduate student at the University of Maine, working in Dr. Melissa McGinnis's lab. And today I'm going to talk to you as well about my research examining how a human-specific virus can cause an incurable and fatal disease in the brain, using large data sets to answer numerous questions. So, as alluded to, JC polyomavirus persistently infects our kidneys of up to 80% of the adult individuals. However, during immunosuppression, the virus can reactivate and spread to the brain, where it causes the fatal demyelinating disease, progressive multifocal leukoencephalopathy. Now, within the central nervous system, it's a very dynamic location. You have neurons that transmit electrical signals and many cell types that support those neurons. When the virus arrives in the central nervous system, one of the main cell types it targets are oligodendrocytes, and the destruction of these cells leads to the hallmark pathology that we observe in PML. But recent evidence has suggested that astrocytes are actually main targets of viral infection. However, infection is poorly understood in this cell type. So my work tries to understand JC polyomavirus infection in a primary normal human astrocyte cell line, comparing infection to a more well-known tool known as SVGAs. However, due to their transformed and mixed population of brain cells, they do not accurately portray viral infection in astrocytes. To broadly understand JC polyomavirus infection in both cell types, I use RNA sequencing, and here I'm going to represent the data as an MA plot. Briefly, MA plots are essentially scatter plots that plot genes that look at genes that were either highly turned on during viral infection, or genes that were downregulated during viral infection, or essentially genes that were differentially expressed. At 1 day post-infection, the majority of genes that were differentially expressed were significantly turned on. However, as JC polyomavirus infection continued or ensued, you began to observe this temporal regulation of genes being turned on and turned off. To compare this to SVGAs, and what we observe here is a distinct temporal regulation that was observed in primary human astrocytes that was not observed in infection during SVGAs, highlighting the importance of using this primary cell line. But bioinformatics can also validate and reveal novel pathways in primary astrocytes during infection. One of those pathways is the MAP-ERK pathway, important in cell survival, cell growth, and cell division, but it may also contribute to the differential gene expression that we observed in the previous plot. Performing something called an overrepresentation analysis, these genes on the left interact with other genes in the MAP-ERK pathway. The genes that are colored yellow or orange are significantly turned on in primary human astrocytes that were not turned on during SVGAs. And what we can do with this analysis is connect these genes with other known pathways within the cell known as a go-cord plot. What this plot revealed is that there are additional pathways in the cell that are important. Here, the astrocytes are activating an immune response to try to combat the virus, but simultaneously, the virus is trying to transform the cell into a virus factory. In summary, RNA sequencing revealed a distinct temporal regulation in primary astrocytes. It highlighted this perpetual fight between virus and host. And most importantly, what we can do with this analysis is target specific pathways involved in JC polyomavirus infection that can be potential therapeutic targets for PML. Thank you.
Thank you so much, Michael. And now we'll open it up to the judges.
Michael, do you see yourself also trying to use this methodology to apply it to other viral infections? In other words, do you want to see whether or not you'd get the same sort of pathways that you've elucidated here?
Yeah, so there are other, they're the same family. They are polyomaviruses, but another polyomavirus that we use in the lab is SV40. And this SV40 is actually used as a negative control as it doesn't use the MAP-ERK pathway during viral infection. But astrocytes are definitely important, especially that before this pandemic, Zika virus was a large problem. And that's when a lot of people started examining astrocytes and how important they were in combating viral infection. So,
okay. So you think that it might be a more generalized mechanism?
Possibly. The analyses that I haven't showed here too is that we're understanding more what astrocytes do in viral infection. We know they activate an immune response. So here, what's really interesting too is that astrocytes are actually possibly activating an immune response to JC infection as well.
Ah, okay, very good. Thank you.
Mm-hmm.
And some of the other responses that you show here, like for example, the gonadotropin releasing hormone receptor pathway, what do you know sort of why that might be, that one is sort of being upregulated?
To be honest, not about that one. So I've looked at all of the other pathways, such as the apoptosis signaling pathway, the gonadotropin releasing hormone receptor pathway. I guess this is where bioinformatics is very useful. But if it doesn't have, we have to think about both the bioinformatic end and the biological end. So even though it's potentially bioinformatics significant, I don't think here it's biologically relevant. So I guess that's the whole point of validating after we find these bioinformatic findings.
Okay, sounds like we're done with questions. Thank you so much, Michael. And thank you to all of our contestants. That concludes the competition part. Just a couple announcements. So for you guys, the contestants, the judges will be deliberating today, and we will be emailing all of you tomorrow, probably around noonish. And then for all of our guests, thank you so much for joining us today. We really appreciate your support and your presence today here. And probably next week, we'll have some more official announcements on our website. We'll probably throw the recording in on our YouTube channel. So if you're curious to know who placed, be sure to check our website. And also if you're subscribed to our email, it'll be there as well. Once again, thank you so much. If anybody has any questions, please follow up via email. And I wish you good luck and have a wonderful evening. Thanks so much.
Thank you.
And thank you, judges, again.